Or maybe you should open your microphone.
Just one sec. Okay. Thanks a lot for the introduction, and also thank you very much for this opportunity to share with you some of the things that we have been doing in my lab in the past few years. I actually like to begin my presentations with this slide because I think it gives a very nice visual summary of what the main research interests in my lab are. Because what you can see here is two tomato plants which are genetically identical and which have been grown in exactly the same environmental conditions. So basically side by side in the growth room. But you can easily notice that they look rather different. So the plant on the right is smaller in size, it's yellowish in color, the upper leaves are curled, and most importantly, this plant on the right will most likely never set fruit, so it will never give us tomatoes. And because you have already seen the title of my talk, then you can probably make an educated guess as to what is happening to this plant on the right. And this plant is indeed infected by a virus. And this virus is completely rewiring plant development and physiology in order to promote its own multiplication and spread. And personally, I think this is fascinating per se, but I hope that you will agree with me that this is even more striking if we take into account genome sizes. Because while the plant that is being infected here has tens of thousands of genes, the virus that is infecting it has a grand total of 6. And yet, with 6 genes, it is perfectly capable of turning a plant like this one into a plant like this one. So just to depict this more visually, I have here drawn two circles, one representing the tomato and one representing the virus. And the size of the circles is proportional to the number of genes that each of them have. And it might be difficult to see, but I swear to you, there are two circles there. And I have used an arrowhead here to make it easier for you to find the viral circle. So you might be wondering how on earth is it possible that this tiny, tiny virus can create such havoc in its host plant? And this is exactly what we are wondering too. So in my lab, we work mostly on one family of plant viruses, and these are geminiviruses. So geminiviruses are fascinating, that they are also relevant. They are causal agents of devastating diseases in temperate and tropical regions of the world. They infect most major cash and staple crops, and therefore they have both economic and humanitarian relevance. And once the plants get infected, they frequently develop symptoms as spectacular as the ones that you can see in these pictures here. So geminiviruses are encapsulated in these beautiful geminate capsids that give the family its name. The viral genome is inside this capsid. It is a circular single-stranded DNA genome. And actually, you can see here a representation of the genome of tomato yellow leaf curl virus, or TYLCV, which has so far been our favorite model Gemini virus, and it is also the virus infecting the tomato plant on the previous slide. So as you can see, the TYLCV genome contains bidirectional and partially overlapping open reading frames, 6 in total, which are here depicted as arrows. So geminiviruses are transmitted by insect vectors. In the case of TYLCV, that is the whitefly, Bemisia tabaci. So when the whitefly feeds into a plant while carrying the virus, this virus has to be released, and then the viral genome has to somehow reach the nucleus. And here, germiniviral DNA replication will ensue. So this single-stranded DNA molecule has to be converted into a double-stranded DNA intermediate, from which, on one hand, viral genes will be expressed, and which will also serve as the template for rolling circle replication. This will produce new copies of this single-stranded DNA genome. Then, of course, the virus has to move both from cell to cell and also systemically in the plant. And all throughout the viral infection, the virus also must suppress antiviral defense mechanisms, which happen at different levels and in different subcellular compartments. And if this viral infection is successful, then there will be the development of symptoms. In my lab, since we started in 2015, so for the past 6 years and a half or so, we have been studying what the 6 proteins encoded by TYLCV do during the viral conquer of the plant. And we have found a number of things for each of these proteins. I'm not going to go too much into detail because most of these findings are already published. But my point is we have learned a bit about how the virus manipulates the plant cell. But I would like to mention that we have also learned along the way quite a few things about plants themselves. And that is because viruses make excellent probes for plant functions. But I would like to tell you a little bit more about 2 different aspects of the viral infection that we have been studying. One of them is viral replication, which involves 2 viral proteins, namely Rep and C3. And the other aspect is symptom development, which is actually carried away by the C4 process. So we are really interested in viral replication because this is the first and most crucial step in the viral infection. And also because it is at this point when variability is generated. And this variability is what in the end allows the viral to adapt, including the breakdown of resistance in the field. So we have been able to identify the elusive DNA polymerases mediating replication of these viruses. And we have found that it is actually the primase DNA polymerase alpha, which initiates DNA replication in the plant, that mediates this step from the single-stranded DNA form to the double-stranded DNA intermediate. And then it is one of the nuclear processive DNA polymerases, DNA polymerase delta, which triggers the rolling circle replication. And interestingly, the other nuclear processive DNA polymerase epsilon, actually has a negative effect in this process. And we can also position the 2 viral proteins involved in this process, namely Rep and C3, in this schematic. So Rep is essential to initiate rolling circle replication, but it is fully expendable for this initial step, while C3 acts selectively recruiting DNA polymerase delta over the non-functional DNA polymerase epsilon. And we have found that C3 and Rep actually interact in the plant nucleus in these beautiful speckles, of which the identity we don't know yet. And what we are now doing is we are using turbo ID-based proximity labeling to try to identify plant proteins that are essential for the replication of the virus. And luckily for us, both Rep and C3 fused to turbo ID are replication competent. So we are using both of them to identify plant proteins that are close to the viral replisome during the viral DNA replication. So this is an ongoing project, but I can tell you that we have already identified a number of plant factors that are essential for this process. And we really hope that this will pave the way for the generation of resistance in the future. And now I'm going to change gears and tell you a bit more about symptoms. And for this, I'm going to introduce the C4 protein, which is, I think, a very interesting viral protein, although, of course, I might be biased. So this C4 protein is the smallest protein encoded by geminiviruses, and it is encoded by this small operating frame here, which, as you can see, fully overlaps with that encoding the replication-associated protein. But I mean, despite the constrictions that this overlap might entail, C4 is the most aberrant protein in geminiviruses. It also contains different combinations of targeting signals in different species, and you can see that on this phylogenetic tree on the left, where these circles represent the presence of different targeting signals, namely a chloroplast transit peptide and 2 different lipidations, meristylation and palmitoylation. C4 is also the only viral protein that is consistently subjected to positive selection. And importantly, C4 is the symptom determinant. That is, it is the viral protein that leads to the development of symptoms during the viral infection. So work in my lab for the past few years have led us to propose this model for the function of C4 in the pathogenesis of TYLCV. So C4 is produced inside the plant cell, and then it is meristylated co-translationally by NMT1, the plant meristyle transferase. And then it basically associates to the plasma membrane where it can interact with a number of receptor-like kinases. And these receptor-like kinases include the 2 close homologs BAM1 and BAM2. So through this physical interaction with the kinase domain of BAM1 and BAM2, C4 can suppress the intercellular movement of RNA silencing. And you can see here on the right the subcellular localization of C4-GFP, which is mostly plasma membrane. And here you can visualize the interaction between C4 and BAM1 by molecular fluorescence complementation or biopsy. So you can see this interaction is happening at the plasma membrane, but it is particularly intense at plasmodesmatar. Now, we have also found that when defense is activated, and this is something that happens when the virus is infecting the cell, then C4 can somehow be released from the plasma membrane and relocalize to chloroplasts. And in chloroplasts, C4 will be interacting with another plant protein, Cas, or calcium sensing receptor. And this will lead to the inhibition of the downstream activation of salicylic acid mediated defenses, which play a very important antiviral role. And what you can see in this last picture here is how C4, when the cell is infected, actually starts being depleted from the plasma membrane and strongly accumulating in chloroplast. So we think this is a beautiful example of how subcellular compartmentalization can enable multifunctionality of a viral protein. But in a nutshell, what I am telling you here is that C4 can suppress plant defense, and it can do so at different levels. However, this does not really tell us anything about how symptoms arise. So how is C4 triggering this symptom development during the viral infection? What we knew was that C4 has to be at the plasma membrane in order to trigger this effect. If we express the wild-type version of C4, which is mostly at the plasma membrane, in transgenic tomato or Arabidopsis plants, you can see that these plants display these very strong developmental alterations that resemble the symptoms during the infection. Now, if we express a non-myristically labeled mutant version of C4, the C4D2A version, that localizes to chloroplast exclusively, then the plants look rather wild-type-like. So C4 has to be at the plasma membrane for the symptoms to appear. So following the rationale that C4 is most likely exerting this effect through the interaction with another plant protein, we decided to do a yeast-2 hybrid screen. And in this yeast-2 hybrid screen, we identified a family, members of a gene family in Arabidopsis that is called the RLD family. There are 9 RLDs in Arabidopsis, but only RLDs 1 to 4 are expressed in vegetative tissues. And I have to say this is a plant-specific family. These RLDs 1 to 4 are precisely the orthologs of the RLDs that we found from tomato as interactors of C4. These RLD proteins are rather large proteins. They are more than 1,000 amino acids, and they contain a number of different motifs. And these motifs include a brevis radix or BRX motif at the C-terminal part of the protein. And because we got a very decent number of independent clones, this interaction came already mapped, and we know that it is precisely this BRX domain at the part of the protein that interacts with C4. But first things first, we confirmed that C4 can interact with the RLDs 1 to 4 in yeast-2 hybrid, biopsy, and Fred-frame assays. But who are these RLD proteins? So the RLDs were first identified by Furutani et al. As interactors of the regulator of gravitropic responses, LACY. So in basal conditions, these RLD proteins are present in intracellular puncta, but when LACY proteins are co-expressed, then they are capable of recruiting the RLD proteins to the plasma membrane through physical interaction. So this leads to polar oxygen transport and ultimately normal gravitropic responses upon gravity-stimulation. Earlier this year, there was a second paper on the function of the RLD proteins, where Wang et al. Described that the RLDs are actually regulators of endomembrane trafficking and therefore general cell polarity. And therefore, this expands the function of the RLD proteins beyond gravitropic responses. In Nicotiana methamiana, the RLD proteins also show a similar localization to that described in Arabidopsis, so they are present in intracellular structures, but this localization drastically changes if we co-express C4. Because C4, just as LACY does, is capable of recruiting the RLDs to the plasma membrane through protein-protein interaction. And interestingly, C4 can actually outcompete the endogenous interactor LACY for RLD binding. So you can see that in this Fred film assay here, so the interaction between C4 and RLD3 is not affected by the presence of LACY, but on the contrary, the interaction between LACY and RLD3, which we can detect here, is completely abolished if we add C4 to the equation. And I will not go into detail for the sake of time, but I can tell you that the interaction between C4 and the RLDs depend on the BRX domain on the RLD side, and on this domain here on the C4 side. So this domain, as you can see, is very similar to the described CCL domain in LACY proteins, which is indeed the domain that mediates the interaction with the RLDs here as well. And we refer to this domain in C4 as the CCL-like domain. So in a nutshell, C4 recruits RLD proteins to the plasma membrane, and it can outcompete endogenous RLD interactors for RLD binding. And this interaction between C4 and the RLDs requires a CCL-like domain in C4. Now, interestingly, this CCL-like domain is required for C4 to cause these developmental alterations. So you can see here transgenic Arabidopsis plants expressing the wild-type version of C4, and they do display the symptom-like alterations. But transgenic plants that express two different versions of CCL-like mutant C4 proteins, they do not display any alteration at all, and they look absolutely wild-type-like all throughout the plant cycle. So this CCL-like domain in C4 is essential for the protein to trigger developmental alterations. And again, I don't have time to go into detail, but just as a snapshot, I can tell you that what is happening here, that is not happening here, is that the wild-type version of C4 can interfere with endomembrane trafficking, which we can visualize because of the appearance of these BFA body-like structures in the plant cells. And this does not happen when the C4 protein does not contain the CCL-like domain anymore. And these structures resemble the structures that appear in an RLD quadruple mutant, and therefore suggest that C4 is interfering with the function of the RLDs through this protein-protein interaction. Now, in order to understand how the CCL-like domain and the interaction with the RLDs contribute to the viral infection, we generated a mutant version of the virus that expresses a CCL-like domain deficient version of C4. So here you can see tomato plants that have been inoculated with a wild-type version of C4, and these plants develop the typical symptoms. With a C4 null mutant version of the virus, and these plants do not develop any symptoms because this virus can barely infect. And plants that have been inoculated with this CCL-like domain deficient C4 mutant virus, and these plants do not display any developmental alterations either, so there are no symptoms. Now, to our surprise, when we measure viral accumulation with these plants, we find that despite the complete absence of symptoms, viral accumulation is wild-type-like for this mutant virus. So basically, in this pathosystem, symptom development and viral performance in the plant can be completely uncoupled. So this is the model that we are working on at the moment. So we think in basal conditions, the RLD proteins are interacting with their endogenous partners and performing their function in the regulation of endomembrane trafficking, and this leads to normal plant development. When the cell is infected, then the C4 protein will be recruiting the RLDs to the plasma membrane and outcompeting the endogenous interactors, and therefore the RLD function will be distorted. This will lead to the development of symptoms, and at the same time, the virus will accumulate happily. And these symptoms that appear during the infection might or might not favor insect transmission. Now, if we artificially inoculate the plants with a CCL-like domain deficient mutant virus, then the C4 protein will no longer be able to interact with the RLDs, which will therefore continue performing their normal function. These plants will not develop any symptoms, but the virus can still accumulate normally. And again, this absence of symptoms might or might not entail a penalty for insect transmission. So I have been mentioning quite some different functions of the C4 protein, and now I would like to share with you what we have learned along the way with regards to how this information is encoded within the protein. So this C4 protein is 97 amino acid in size, and we know that there is a meristylation site in position 2. This meristylation site is essential for plasma membrane localization, and therefore for the suppression of the intercellular movement of RNA silencing, and also for symptom determination. Then we have the chloroplast transit peptide that spans the first 44 amino acids of the protein. And this part of the protein is, of course, essential for chloroplast targeting, and therefore for the suppression of salicylic acid-mediated defenses. But this is also the part of the protein that interacts physically with BAM1, and therefore it is required for the suppression of intercellular movement of RNA silencing. Then we have the CCL-like domain, spanning amino acids 52 to 63. And as I just showed you, this is the region of the protein that interacts with the RLDs, and therefore is essential for symptom development. And then we have the rest of the protein, which is the part that interacts with Cas in the chloroplast, and therefore it is essential for the suppression of vesicle-mediated defenses. And if you are not blown away by how compact this spatial information is in the protein, then I will remind you that C4 is encoded by this open reading frame that is fully overlapping with this other open reading frame, but in a different reading frame. And actually, we are probably not done with C4 yet. We have recently found that C4 can physically interact with another viral protein, which is V2, and this interaction happens in intracellular puncta, some of which seem to be plasmodesmata. So this is interesting because it has been long discussed in the field whether C4 or V2 might actually might be exerting a function in viral movement, and this result raises the possibility that perhaps both viral proteins actually act together to mediate the cell-to-cell movement of the virus. And interestingly, this interaction between two viral proteins that we see with C4 and V2 seems to be the rule rather than the exception. So we have found that the 6 canonical proteins encoded by TYLCV interact with one another in a very intricate network, as you can see here, and this can be confirmed by a number of different protein-protein interaction techniques. Now we have taken one of these protein-protein interactions, that between C2 and CP, as a proof of concept, and we have found that CP is sufficient to modify the subcellular localization of C2, and also to completely modify the changes to the transcriptional landscape that C2 triggers. And this result raises the idea that combination of viral proteins might actually enable novel functions, and therefore these interactions may expand the functional landscape of the viral proteome. This gets even more complex if we take into account that we, in collaboration with Shui-Ping Zhou and Fan Li, as well as others, have recently described that there are novel proteins encoded in viral genomes. These novel proteins can be detected during the infection, and they display specific subcellular localizations. And I think that it is very possible that geminiviral proteomes have been underestimated by at least 50%. So all things considered, I think that what we know about geminiviruses right now amounts to this much. And therefore, we have our work cut out for us. And with this, I would like to thank the people in my work. I would like to say a big thank you to the past and present members of my lab, because without them, none of this would be possible. I would like to thank our fantastic collaborators and, of course, funding agencies, and you for your attention.
Thank you, Rosa. Thanks for your interesting talk. And now is Q&A session. Committee members, if you have questions for Rosa, please feel free to open your microphone.
Hi, Rosa. Very nice talk. I'd like to ask, does the interaction of C4 with BAM1 affect symptom development, given that BAM1 is also a meristem gene? And also, does BAM1 interact with RLD?
Yeah, so with regards to the first question, we know that the CCL domain mutant, CCL-like domain mutant version of C4 can still interact with PAM1, and these plants are completely symptomless. And I mean, in agreement with our model, actually, these mutant versions of C4 can still impair the cell-to-cell movement of silencing. So the interaction with PAM1 and the associated function are maintained, but the symptoms are gone. So we think that the PAMs do not play any role in symptom development. And we don't know whether the PAMs interact with the RLDs. We have never tested this.
Okay, thanks.
Thank you.
So Rosa, you're talking about intermolecular interaction among proteins encoded by the viral genome. Did I catch that right?
Yeah,
at the
perfectly
last
right.
minute. Yeah.
Yes.
So you're saying there may be gain of function from this kind of interaction. Do you mean it's like in the context of host effect or in the context of virulence of the virus?
Sorry, in the context of what? I missed the last part of the sentence.
The virulence of the virus.
So, I mean, with the data we have so far, we think that some of these interactions might be essential for viral performance. So, for example, it has been described before that the rep protein has to homodimerize. And if this doesn't happen, the replication just doesn't occur. And in the case of the interaction that we have been studying, C2 and CP, CP can completely change the interaction of C2. So C2 now is present in the nucleolus where it is not present in the absence of CP. And this really affects the transcriptional response of the plant, or the transcriptional changes that are concomitant to the expression of C2. And this is not, so C2 alone can trigger very strong changes in transcription. CP alone doesn't affect transcription at all. But if you put them together, then the transcriptional changes just are modified. They are not an addition to what C2 does, but it's something new. And we think this could actually have an impact during the infection.
Thanks.
Hi, Rosa. Very nice talk. In your talk, you show that C4 of this virus TYLCV is very important for induce the development phenotype. So I'm just wondering. Because this virus TYLCV can only affect some plant species. Did you look at? Did you test this virus can infect monocot plant species like the rice or maize? So did you put the the C4 into the rice or maize to try to see there's only developmental phenotype?
Yeah, we haven't done this. So actually, TYLCV is restricted to dicots, mostly Solanaceae. There are some geminiviruses infecting monocots, but these do not seem to have C4 proteins. But in agreement with this, the symptoms are quite different, actually. So we have not expressed C4 in monocots. I think this would be something interesting to do. My gut feeling would be symptoms would also be developed, like these developmental symptom-like developmental alterations. So one example is, for example, in the case of Arabidopsis, so TYLCV does not really infect Arabidopsis very well. So it can infect, but the infection is symptomless in Arabidopsis. And yet when we express C4, we see these effects in development very clearly.
Okay, thank you.
Thank you.
Thank you, Rosa. Thanks again for your nice talk. Now all the 10 finalists have delivered their wonderful talks. And thanks to everyone for making RSPS 2022 an extraordinary journey to plant science. And last but not least, please join me to welcome Professor Shen Luan from the University of California, Berkeley, and also Editor-in-Chief of Molecular Plant, to make a concluding remark. Please, Professor Luan.
Hello, hello. Hi, everyone. I'm sure we have all enjoyed the outstanding talks by our rising stars in plant sciences. As you know, we're celebrating our 15th anniversary for Molecular Plant, and also our recent establishment of the sister journal, Plant Communications. Through the years, we have been always following our initial mission. That is, we wanted not only to establish leading journals in plant science, but also build platforms and bridges for communication and collaboration among all researchers in the international community. So this rising